I warmly welcome you and thank you in advance for being here in this afternoon. If you are looking for Lise, I'm sorry to disappoint you. Due to family circumstances, she's not able to be here, so I am not Lise. I am Pennadam Sivanand, and I will walk you through today the particulates, endotoxins, and bioburden characterization of silicone tubing for biopharmaceutical applications. Before we dive into the technical details, I would like to show you the context of the presentation and just quickly show you what I am going to talk about. And we will just briefly go through Dow Forte, which is strength, and our capabilities, Dow Pharma Solutions, and we will briefly cover about Dow Corning Pharma tubing offerings that we have in biopharmaceutical, and what is the market requirement there, and the much desired or interested topic, which is the testing protocol on data. And we will cover those 3 testings that we have done and with a slide summary of conclusions. So first about Dow Forte and its capability. As you are all familiar, Dow is a well-known chemical company with over 165 years of diverse experience in delivering award-winning technologies in collaboration with our customers, where we develop products, used our innovation capabilities, which is directly desired to address the customer needs. Thanks to our marketing colleagues and the insight they bring on the market trends where the consumers are going forward with the needs, we develop innovations. Dow is committed to healthcare, and we have a broad range of, We have a broad range of material science expertise, and our product and application know-how combined with the reliable quality and the service that we provide is welcomed by our customers and supported. We also support this with our regulatory experience, with detailed product knowledge and capacity. I also want to highlight our capabilities. We span across the world with at least 150 employees focusing particularly on R&D and product development. And our products are manufactured in 9 different locations, which helps our customers buy from at least 90 countries or over. And the R&D colleagues are spanned across in 6 different regions in locations across the globe. Okay. And going into Dow Pharma Solutions, for the past 18 months, we are fully owned and subsidiaries operated under Dow Chemical Company. So we have integrated Dow Silicones and the Dow Organic with the combined expertise of organic and silicone. We are able to provide market-leading capabilities and expertise to support our customers' growth. And we have diverse product portfolio range with silicones and organic that span across the entire range, which you will see in a couple of slides. Our innovations helps provide new treatment and flexibility for patients, which enhances the life quality, living healthier longer. As I mentioned in the previous slide, combined organic and silicones, we have a broad range of products offering to our customers. I will briefly talk about certain elements that we play a role in. Under the API section, as you can see on the left-hand side of the screen, we play gastroenterology, hyperkalemia, and hypercholesterolemia. And on the topical excipients ingredient space, we operate in various segments. In topical space, we have a broad range of silicone products and organic products that helps in ingredient and excipients, which not only provides aesthetic benefits and also gives sensory performance, and most importantly helps drugs stabilize and deliver topically. In the liquid space, our product help control release, taste masking, solubility enhancement, and the solubility improvement of the products of our customers. In the tablet space, we have, again, solubility enhancement combined with sustained release or delayed release or immediate release, depends on the need of the material. In the transdermal, I want to emphasize we have products that has got standard BIOPH elements and amine-compatible BIOPH elements, which combined broadly offers the ability to customers to tailor-made the API that needs for the applications. On the capsule space, we have solubility enhancement of our material that offers immediate release of the drugs. And we also play a mild role in the mucosal bioadhesives and suppositories. And most importantly, which is relevant to this presentation, is where we play on the process side in the biopharmaceutical processing, our tubing. So we have medical grade tubing and pharma tubing. Along with that, we also play a major role in the overmolded assemblies. So Just give you a brief idea about Dow Corning Pharma tubing. Silicone tubing is well known and it's used in the fluids transportation specifications, and particularly targeting biopharmaceutical and biotechnology applications, including, not limited to, drug production, aseptic processing, fill, finish and packaging, and most importantly, in all the single-use systems. I would like to emphasize that all our Dow Corning silicone tubings are produced from Silastic biomedical grade elastomer, which employs a bespoke, niche, platinum catalyzed addition Q technology. It offers many customizable options, and it is currently available in 3 durometers, such as 50 Shore A, which represent to the strength in Armstrong units, 65 and 80. As we go higher, the strength, the flexibility of the tubing differs according to the need. So if I just briefly take you through the chemistry, as I mentioned, it is using a bespoke niche silicone platinum catalyzed Q technology, addition Q technology. So as you can see on the top left-hand side or right-hand side of the screen, it employs vinyl polydimethoxyl silane. Unfortunately, the laser won't work. So if I just brief you to the circle point where you have a vinyl silane, PDMS, combined with silyl hydrate with small circle emphasizing the hydrogen point of the silicone. So in presence of the platinum catalyst, the hydrogen replaces to the double bond, and which gives you the final product, which is your cross-linked elastomer. Okay. So the way it works is we use base siloxane polymers with siloxane cross-linkers in presence of filler silica, which uses to reinforce the material. And with the catalyst, we have a cross-linked network system. Okay, so yeah. So as you can see, the chemistry doesn't employ any processing aid, neither organic plasticizers or stabilizers. Thus, we have no byproducts. What it does mean is it gives you low extractable profiles in your final application tubing space. Okay. So what is the benefits of the silicone tubing? It's first and most importantly safe and secure. When I mean secure, it's secure for our customers. How? It provides cleanliness in use. That means it has got low ability to particle entrapment and low biofilm adhesion that prevents organism growth. It is biocompatible, meaning it's inert. It has got no adverse effect on human health. Example, skin or eye irritation, genitotoxicity, and finally but not least, it's pyrogen tested. It comes with high purity, as you have seen in the previous screen or slide, that it doesn't have any other added elements to it, and it is easy to sterilize. We, as Dow Corning silicone products and pharma tubing, have a long proven history in the field, and our integrated supply chain is no one can match, and which you can see in the next slide what I mean on that. Starting from quartz all the way up to the tubing, or including our molded assemblies or over molded assemblies, we have complete control on every single step of manufacturing. So combined or compared to our silicone competitors, they may able to claim up to certain part they have the same integration, and definitely our tubing competitors could only claim they have the vertical integration only on the last two factors. So given that fact, as you can see on the highlight, we have complete traceability starting from the end product, supply chain quality control, integrated change control with the reliability of supply, which is very critical for our customers like you. So what is the market requirement? Based on what we understood from our customers as well as our market insights, on the high purity requirements for biopharmaceutical processing is ever increasing. Because silicone, sorry, I lost the mic, yeah, thanks. Because silicone tubing provides either a standalone or as a single-use system component in biopharmaceutical downstream manufacturing process. The critical downstream manufacturing steps in production or parenteral drugs of proteins, vaccines, and final filtration, fill finish, and packaging, etc. require avoid foreign matters, endotoxins, microorganisms, and the contamination of final drug product. So customers have increased requirements for testing the purity of the single-use systems and components with regards to purity from suppliers such as us. And so they expect to provide us in support of their application final requirements. offer the testing on extractables and other three that I have highlighted. The reason I haven't highlighted the extractables, our product is well known for its extractable testing. So now with the new testing that we have completed, we could offer a complete package of data that covers all the requirement testing with the regulatory support. So what have we tested and on what product we have tested is listed here. We have committed to help the pharma industry in reducing risk from the supply chain. Consequently, we developed the most appropriate and stringent tubing-specific testing protocol and providing a supreme data package. We have tested the entire range of our Dow Corning product family, such as 50 pharma tubing, 65 Shore tubing, and pharma 80 tubing, along with the pharma advanced pump tubing. The way we have done it, all the testing have been executed in multiple batches and in various sizes of tubing, so not limited to certain specifics. And all the testings in endotoxins, we have done internally in our Dow QC lab in States last year. So to eliminate the bias of internal testing, we have reached out our collaborators or laboratories that is certified to carry out these testing across the world. As you can see, particulate testings were carried out in States and bioburden were done in Europe. So this could give a broad confidence in the data package that we developed to our customers who had used globally. So what is the visible or subvisible particulate testing? As you all can understand, subvisible is something that you can't see through your naked eye. So it talks about anything under 100 micron lesser range on the left-hand side of the top screen. Just wanted to give you a quick highlight on what is the definition of particulate. There are two classifications of particulates according to USB and BPSA. As you can see, USB 788 very clearly defines anything that is extraneous, mobile, undissolved particles other than gas bubbles, which is unintentionally present in the solutions is called particulates. According to BPSA, again, the mobile or embedded matter unintentionally present in the single-use component assembly may contact or end up in the process or product fluid. So we have taken the USB 78 to determine our particulates in the tubing, and we have gone one of the critical method. As you can see on the bottom of the screen, there are two methods. One is light abstraction method and another is membrane microscopy method. So Dow, being a leader in the pharma tubing, had gone to the stringent testing criteria of the membrane microscopy, which you will see in the next step how we demonstrate. So here there are passing criteria that I would like to highlight. So anything the volume over than 100 milliliters in the production. According to the membrane microscopy method, there should be less than 12 count per ml for particle size that is greater than 10 micron but less than 25 micron. But if you talk about volume of 25 micron particulates, you should have less than 2 counts per ml. So that is your criteria for passing in the tubing. Let's see how we do on the testing. Okay, the testing was done on the solid particulates I mentioned in the previous slide. So we have taken the tubing, filled it with our material that would potentially extract all the particulates. So the eluent that we used is as particulate-free water. All the extracted volume were pooled together, collected, and filtered on a membrane for enumeration. This membrane then gone through the microscopic count acceptance criteria to see how much particulates are there in our products. So as you can see in the screen, I again wanted to emphasize on the limit or expectations. So we should have at least 12 counts or less than 2 counts for the particle size 10 microns or 25 microns. So if you look at our range of pharma tubing that's gone through this testing, it's very evident we have no more than 0.56 on the particle size of 10 micron or above, less than 25 micron, and we definitely have no more than 0.2 count for the particle size of 25 micron to 100 micron. So this clearly illustrates on our stringent criteria, we pass in flying colors of the particulates that is in our tubing or expected in our tubing. So that gives a very much confidence for our customers, how they can use with reliability and ease of use in this field. So I wanted to take you to the next one, which is our bacterial endotoxin testing. So I would like to emphasize again, endotoxins are pyrogen components of the lipid polysaccharides layer of gram-negative bacterial cell walls. So we have taken here the USP85 bacterial endotoxin test, which employs limulus amoebocyte lysate, which is used to detect the quantify bacterial endotoxin from material sample. So there are two types of techniques. One is called gel clot, where the detection of endotoxin based on clotting of the LAL reagent that I specified in the above, that shows the presence of your endotoxin. The another one is photometric method, which provides you turbidity and chromogenic techniques employs. So we ruled out the second method just to emphasize there is no limitation with the analytical techniques that we use by the chromogenics or turbidity measurements. So we went for the very clear visible gel clot, which employ another stringent criteria of 0.125 endotoxin unit per ml sensitivity. So here you can see the tubing extract is exposed to this agent, and then see if there is a gel formed. If there is a gel formed, it fails. If there is no gel formed, it passes the criteria. So as you can see again in the screen, it's very clear. All our range of pharma tubing tested has passed with flying colors. Again, taking to the limits of USP, we are in the operation of 0.125, but according to the USP that we followed, we would like to have at least 0.25, but we are less than half of what is required. So again, this gives confidence to our customers and consumers that our tubing has got very high purity and high standards in the biopharmaceutical field that we employ. Finally, not but least, the bioburden testing. So here we employed the ISO 11737 based on our expertise in the medical device field that we operate, and we have used the microbiological methods part 1. It counts the population of microorganisms in the products. It can be done in either just aerobic bacteria alone or in combination with yeast and molds. But most critically, what we have done is we have gone for the full range of microbials, which is bacteria, yeast, mold, and spores. The first thing we need to understand and gain confidence on the validation, how we are confident in doing the inoculation method and how we are recovering the particulates. So we had repeated experiments and we have complete confidence on our validation. And so we considered also the correlation factor that goes into, because the way the testing was done is after the evaluation, you dilute the product in order to do the further testing. So the dilution factor needs to be employed and make sure that it applies the correlation factor correctly. So what I have mentioned is after removal of the microorganism from the test material, the elutin is membrane filtered and the filter is incubated and counted for the bacteria. Again, as you can see on the screen, all our products for every single units that we measured, let it be aerobic, yeast, molds or spores, we have none detected. So there is minimum that you could say 1 or less than 1, including the diluting factor. There is nothing detected in our tubing, which is really, again, reassuring and emphasize the point that the pharma tubing from Dow Corning is at the highest purity and the limits. Okay, just wanted to summarize based on our 70 years expertise in material science, definitely over 50 years expertise in healthcare materials. We offer testing protocol development and data dossiers that support our leadership in high purity solutions and accelerate the validation process of our products for biopharmaceutical manufacturing. Our unique vertically integrated supply chain allows every step of the manufacturing process to be accounted for and stay under our control. Our pharma tubing products are meeting and exceeding the requirements of the USP and ISO regulations. Particulate counts are sustainably lower than the acceptance limits of USP 788. USP 85 says bacterial endotoxin level is below our stringent 0.125 endotoxin unit per ml acceptance limit. No bioburdens was detected as per the ISO 11737 method. These characteristics make Dow Pharma tubing particularly suitable for critical biopharmaceutical process where purity is key. I would like you to invite to visit us in our booth at 10.2, the booth number 102A50, to learn more about our range of product in pharma solutions, and to connect with us, collaborate with us to address your unmet need, and to innovate with us for your future requirements. You can also welcome to visit our YouTube. I believe it works still, so please do. And I am more than happy to take any questions that you may have, and I sincerely thank you for your attendance and time.